Following presentation, we look into phages as enemies with specific interest in food fermentation. The main goal of this presentation is to make viewers aware of the relevance and implication of phage attacks in dairy fermentation. Specific emphasis will be given on an introduction to lactic acid bacteria, in short LAB, the association of phages with LAB in terms of phage-host interactions, phages affecting LAB with particular interest to lactococcus, how bacteria have evolved to escape phage infection, and the practical aspects related to this problem in the dairy fermentative industry. Lactic acid bacteria constitute a diverse group of microorganisms associated with different products such as plants, meat, and dairy. The lactics are also important commercially in the processing of meats, alcoholic beverages, and vegetables. They are used in the manufacture of dairy products such as acidophilus milk, yogurt, buttermilk, and cheeses. Lactic acid bacteria comprise different groups of microorganisms such as Canobacterium, Enterococcus, Tetragenococcus, Vacuococcus, and Weissalia. The species of the genera that constitutes the industrial core of LAB include Lactococcus, Lactobacillus, Leuconostoc, Ionococcus, and S. thermophilus. So far, the genus Lactococcus is the best characterized food-related LAB because they are able to grow milk and transform lactose to lactic acid. For this reason, they are commonly used as starter cultures in industrial fermentation for cheese production. As early as the mid-30s from the 20th century, the first negative effect of bacteriophages on the dairy fermentation was reported. Up to date, regardless of sanitation precautions, starter culture rotations, and constant development of new phage-resistant bacterial strains, phages constitute one of the main and economically most serious sources of fermentation failures. Phages induce bacterial cell lysis, which further lead to failed or slow fermentation of milk, decrease in acid production, and reduction in milk product quality, which in effect causes profound economical losses. To further illustrate the detrimental effects of phages in the fermentation industry, an example of a time-lapse microscopy was done, showcasing infection of Lactococcus lactis with virulent phages. Apparent lysis was observed within 1 hour of infection. Different LAB species can be infected with different phages, such as those that belong to Cephuviridae, Podoviridae, and Myoviridae families. However, the most important are the lactococcal phages. They are classified into 10 groups based on their morphology and genetic relatedness. Nonetheless, 3 main species, 936, P335, and C2, dominate the dairy industry in terms of their persistence and frequency of isolation. Members of the 936 and C2 observe a lytic cycle only, while P335 members might propagate lytically or may integrate their genome into that of the host and replicate in tandem with the host's chromosome, a scenario during which they are termed as lysogenic or template phages. How smart have bacteria become to overcome phage infection? Lactic acid bacteria, as well as other bacteria infected by phages, have evolved defense systems against bacteriophages, which allow them to survive in an environment full of their predators, in this case the phages. These antiphage systems have been organized into groups depending on the manner by which they operate. Some of the systems include inhibition of phage attachment and adsorption, blocking of DNA injection, restriction modification systems, and the fourth is abortive infection systems. However, Many of these systems can be found in conjugative plasmids. In the dairy plant, it is important to identify the potential sources of phage contamination and to limit their entry to the fermentation process. Phages can originate from raw milk, which contains the most probable source of naturally occurring virulent phages at titers as low as 10 to the third plaque-forming units, and also constitute a continuous supply of bacteriophages in the dairy plant. Phage concentrations in raw milk also depend on the conditions of collecting, handling and storing of the milk by the supplier, on the transport to the plant, and finally, handling of the milk in the plant itself. It is also important to remember that the biodiversity of phages is increased by combining milk collected from different farms, and these numbers can even be higher in processed milk. Whey protein concentrates are normally used to standardize milk before the fermentation process or to improve the taste and the texture as well as the nutrient value of the final product. Whey protein concentrates can be also sources of high temperature resistant phages and can influence the quality of the final product. Starter cultures itself can be sources of phages when strains contain temperate phages. The main source of lysogenic strains are undefined cultures for two main reasons. One, the exact strain composition of these starters is unknown, and two, elimination of lysogenic strains from undefined culture is very difficult. Temperate phages are incorporated into bacterial chromosome and the genome replicates together with the bacterial genome. Prophages are carried in many LAB strains and may be induced from lysogenic to lytic form by the manufacturing conditions such as heat, salts, acidity, bacteriocins, starvation, or UV. Prophage induction from multiple lysogenic starter culture strains has the potential to influence fermentation. On the equipment and factory environment, phages are important and commonly present on working surfaces and usually need the presence of the bacterial host. Due to this fact, they are usually found in places where conditions for LAB development are favorable, such as valves, crevices, and dead ends of production lines. Sequentially, in the air raw milk handling, cheese milk processed in the open vats, and whey handling can lead to spreading phages. Virulent phages can circulate through the air far away from the aerosolization sources due to their ability to bind to small particles. So what are the strategies used to control phages? This includes cleaning and disinfection, which is an essential part of milk processing. Cleaning in place procedures are usually applied in milk in the processing lines. The cleaning process can remove 90 or more of microorganisms associated with the surface, but cannot kill all of them. The presence of LAB among the residual microorganisms increases phage risk contamination. In the past, starter culture rotations of undefined multi-species and multi-strain cultures was the main strategy to overcome production problems related to phages in many factories. Currently, the rotated strategy of defined multiple strain cultures encompasses selection of strains resistant to the wide range of phages, which could replace infected strains. But it is important to remember that continual rotation of multiple strains during fermentation process has an effect on phage coevolution. Continual rotation can also increase phage diversity and their abundance in the dairy environment. An alternative is to use a single, highly specialized phage-resistant strain and its variants carrying phage-resistance plasmids obtained from naturally resistant strains. Also, an important element during the spread of phages in the dairy plant is the organization of production. The control of phage risk in the dairy plant relies on development and implementation of a variety of procedures such as aseptic technique, closed fats, page inhibitory media, and severe heat treatment of bulk starter media. In conclusion, phages represent a constant threat of serious economic losses in the dairy industry. As a result of this, dairy microbiologists have attempted for the longest time to eliminate, or at least bring under better control, bacteriophages that interfere with the manufacture of fermented milk products. Phages rapidly disseminate in the dairy environment and are difficult to eliminate. Thus, it is important to understand how the phage and the host coexist in order to gain a better understanding of this complex relationship in the hope of finding sustainable solutions to the ongoing problem faced in the dairy industry. Some important references have also been provided highlighting the impact of phages in the fermentation industry.